Welcome to Chapter 2 of our video series, Cell, the Building Block of Life, Part 1. We'll explore the fundamental unit of life and its origins. Let's begin by looking at a hot spring in Puga Valley, Ladakh, an environment that provides clues about the origin of life on Earth. Life likely began in water environments like these hot springs about 3.5 billion years ago, mimicking early Earth conditions. The organisms living here are mostly unicellular, heat-loving bacteria called thermophiles. Calcium carbonate deposits formed around these hot springs may have protected early organic molecules from harmful radiation and extreme conditions, possibly aiding in the formation of the first protective cell membrane. The process shows small molecules in the air, leading to chemical building blocks and self-assembled structures in water, marking the pre-cellular era and the emergence of protocells. Abiogenesis is the gradual process by which life arose from non-living matter through natural chemical and physical processes over 3.7 to 4.2 billion years ago. The abiogenesis hypothesis outlines the steps, from the primordial earth and soup to the synthesis of simple molecules, formation of complex polymers, assembly of protocells, and finally, the first simple cell or prokaryote. Organisms can be unicellular, composed of a single cell like amoeba or bacteria, or multicellular, where cells group together with different functions to form various body parts, as seen in plants and animals. Life organizes from the basic cell level into tissues, then organs, which form organ systems, and ultimately, an entire organism. The minimum distance at which two objects can be seen separately by an optical instrument is the limit of resolution. The human eye's limit of resolution is 0.1 millimeters. This chart illustrates the size range of objects and their visibility, showing that a light microscope can resolve down to about 1 micrometer, and an electron microscope is needed for smaller structures like viruses and molecules. The unaided eye can resolve objects down to 1 millimeter, such as a fish egg. Objects smaller than 0.1 millimeters require a microscope to be seen clearly. In 1665, Robert Hooke examined cork cells under his self-designed microscope. He observed a honeycomb-like structure with small compartments, which he called cells, meaning a little room in Latin. The compound microscope has several parts, including the eyepiece, objective lens, and adjustment knobs. The eyepiece and objective lens determine the total magnification and what can be resolved. To estimate the real size of a cell, first convert the diameter of the visible field to micrometers. Then, divide this diameter by the number of cells counted along the diameter. For example, 10,000 micrometers divided by 25 cells equals 400 micrometers per cell. The total magnification of a microscope is calculated by multiplying the magnifying power of the eyepiece by the objective lens. For example, an eyepiece of 10x and an objective of 10x results in 100 times magnification. Scientists have enhanced microscopes by improving resolution, which is the measure of clarity, contrast, the difference in brightness, and magnification. These improvements make the microscope a powerful tool for studying cells. This is an electron microscope. The first one was developed by German engineer Ernst Ruska in 1931. Electron microscopes utilize a beam of electrons instead of light to produce highly magnified and clear images. They allow us to observe cell structures at the nanometer scale. A nanometer is 1/billionth of a meter. Comparing the images, a light microscope provides a more basic view, while the electron microscope reveals much finer detail and higher magnification of the cell structure. This concludes Part 1 of our video series. Please follow the link to continue with Part 2, Cell, the Building Block of Life.